Welcome to AP Biology. This video is on the Diffusion and Osmosis lab. This lab has three parts to it. Part 1 is on diffusion. Specifically, the goal of Part 1 is to determine how size affects diffusion through a semi-permeable membrane. The semi-permeable membrane that's going to be used in this part of the lab is dialysis tubing. Here you see it in the upper right hand corner. Dialysis tubing is semi-permeable. It contains small holes or pores in it that are going to allow some things to travel through and not others. Dialysis tubing is used to mimic the cell membrane in this lab. Using the dialysis tubing, we're going to mimic a cell. We're going to create a dialysis bag. To create the bag, you're going to tie off one side of the dialysis tubing using a string. You're going to use a funnel to insert into the dialysis tubing our solution. You're then going to tie off the other side using another string, creating this dialysis bag. The dialysis bag is then going to be placed in a beaker of water. The solution inside the dialysis bag is going to contain both glucose and starch. Glucose being relatively small, starch being pretty large. We're going to see if the glucose and starch solution are able to travel out of the bag into the surrounding solution. To test for glucose in the surrounding solution, we're going to use glucose test strips. To test for starch in the surrounding solution, we're going to insert a chemical called IKI. IKI stands for iodine potassium iodine. And this chemical is normally gold in color. If it comes in contact with starch, it turns from gold to a deep purplish color. So it's an easy indicator for the presence of starch. So will glucose be able to diffuse out of the dialysis bag? Will starch be able to diffuse out of the starch bag? These are the questions that we're trying to answer in this part of the lab. You're going to keep the dialysis bag immersed in the water for 30 minutes, and you're going to record your results in table 1.1. Part 2 of the lab is on osmosis. Our goal specifically is to determine the percent change in mass of dialysis bags due to osmosis taking place. You're going to use 6 beakers, and you're going to construct 6 different dialysis bags. Inside each dialysis bag, you're going to place a different molarity solution of sucrose. Sucrose cannot diffuse through the dialysis bag. Only water will move. One bag will contain 0 molar, or pure water. One bag will contain 0.2, one will contain 0.4, 0.6, 0.8, and 1.0. You're going to weigh each bag prior to placing it in the solution. You're going to put it in the beaker of water for 30 minutes. After 30 minutes, you're going to take it out and you're going to reweigh them. After you've weighed each bag, you're going to calculate a percent change in mass, and you're going to graph this data and place it on a graph. Part C investigates water potential, and we've covered water potential in class. The goal of Part C is to determine the molarity of a potato core and its solute potential. We're going to construct these tubes of potato using a tool called a potato core. Again, we're going to use 6 different beakers, and inside each beaker you're going to place 4 potato cores. Prior to putting the potato cores in each beaker, you're going to weigh them. Inside each beaker, you're going to place a different molarity solution. In Part 2, you put the different molarity solutions inside of the dialysis tubing. In Part C, you're going to be putting the different solutions into the beaker itself. Again, you're going to weigh the potatoes before and you're going to weigh the potatoes after, and you're going to determine a percent change in mass. You're going to plot the percent change in mass in a graph, just like you see on the slide right here. On the bottom, on the x-axis, you see the different molarity solutions, and on the y-axis, you see the percent change in mass. Notice that some potato cores will gain mass, some potato cores will lose mass. The potato itself has a particular molarity, and that's what we're trying to figure out. So depending upon whether the potatoes are in a hypertonic or hypotonic solution, they'll either gain or lose mass. You plot your data and you draw a line straight over from where you have a 0% change in mass. You see where that hits your line and you draw a line straight down. At that molarity right there, where there's a 0% change in mass, you have the molarity of your potato cores. Once you have the molarity of your potato cores, you could use that information to determine the solute potential. Here you see what we need to hand in for the diffusion osmosis lab. For each section, you need to write down the purpose, you need to write down the hypothesis, you need to identify the independent and dependent variables. Each experiment needs a different data table and or graph. For experiment 1, you just need your data table. For experiment 2, you need the data table and graph. For experiment 3, you need the data table, the graph, the molarity of the potato core, and the solute potential. Finally, the last thing you have to write up is the conclusion. In the conclusion, you explain the results for each section. Why did we get the results that we obtained? See you in lab.